Okay, so now moving on to the second part of Unit 2, which is the absorption of carbohydrates, proteins, and lipids. Okay, so absorption of food, for that we have to know the layers of the alimentary canal. ठीक है? So mainly alimentary canal is 4 layer আছে. From the inside, the first one is mucosa, then submucosa, then muscularis, and then serosa. ওকে, তো ইনারমোস্ট লেয়ার যেটা লুমেনের ঠিক পাশেই সেটাকে বলা হচ্ছে মিউকোসা। দিস ইজ মেড আপ অফ আ লেয়ার অফ এপিথেলিয়াল সেলস, সেগুলো মেনলি কলামনার এপিথেলিয়াল সেলস হয়। অ্যান্ড দ্য নেক্সট লেয়ার ইজ নোন অ্যাজ সাবমিউকোসা, এখানে ব্লাড অ্যান্ড লিম্ফ ভেসেলস আছে। Next is the muscularis. So muscularis, as the name itself suggests, it has a layer of circular muscles and the outer layer of longitudinal muscle. তাহলে এই দুটো লেয়ার হচ্ছে মাসকিউলারিস, ভেতরের ইনার লেয়ারটা হচ্ছে সার্কুলার মাসেলসের লেয়ার, আর বাইরের আউটার লেয়ারটা হচ্ছে লঙ্গিটিউডিনাল মাসেলের লেয়ার। ওকে, নাও after that the outermost layer is known as serosa, which contains blood, lymph vessels, and also it may contain adipose tissues. তাহলে এই 4টে লেয়ার দিয়ে হচ্ছে আমাদের এলিমেন্টারি ক্যানালের ওয়ালটা তৈরি। From the inside to the outside, 4 layers. Now if we compare the walls of the small intestine with that of the large intestine, then what we will find is that intestinal villi is present only in case of small intestine, and in large intestine the villi is not present. Here you will find only intestinal crypts, which is quite similar to that of the small intestine, but the prominent finger-like projections, which are known as villi, those are absent in case of large intestine, because it's very obvious that the major part of absorption happens in the small intestine only, and the large intestine does not participate in absorption of digested food. Okay. So from here also this layer is the mucosa, then this is the submucosa, and then this is the muscularis, this portion, and then of course the outermost covering is known as serosa. Okay. First and foremost is the absorption of carbohydrates. So we know that the complex carbohydrates like starch, glycogen, they start getting digested in the mouth where salivary amylase is present. Salivary amylase can convert the starch into maltose. Now these carbohydrates, among them only glucose is directly absorbed in the blood, and the chemical digestion or the hydrolysis of carbohydrates occurs in the presence of different enzymes. They are called glycosidases, which means carbohydrate digesting enzymes are known as glycosidases. They are also called amylolytic enzymes. And this process is known as hydrolysis because it involves the water molecule, and this water molecule helps in the breakdown of the carbohydrates. Now when the acidic chyme that I spoke about in the first video, that reaches the stomach, the partially digested starch, that is hydrolyzed by the action of gastric HCl. So when it reaches the stomach, we already know that HCl is present in the stomach, which makes the pH of the stomach acidic. And in this acidic pH, this helps in the protein digestion like pepsin and all. Now what happens to the carbohydrates? The carbohydrates is further digested in the intestine with the help of pancreatic and intestinal amylases. And of course, before that in the stomach with the help of gastric amylase. Now, disaccharides like sucrose, lactose, maltose, they are digested in the intestine by the action of various enzymes called invertase, beta glucosidase, and alpha glucoalactosidase respectively. Now they convert the sucrose to glucose and fructose, lactose to glucose and galactose, and maltose to 2 glucose molecules. Okay. So the absorption of the digested carbohydrates, it takes place through obviously intestinal villi, either by diffusion, because the small molecules can directly enter the villi through diffusion, or it requires active absorption with the help of ATP. Now fructose is, for example, it can be taken up by the simple diffusion process only when there is a concentration gradient. From higher concentration to lower concentration, fructose can be absorbed by the cells of villi. While glucose and galactose, they need to be actively transported with ATP hydrolysis. So if excess glucose is taken up in the diet, then what happens is that the muscle and liver, it stores the additional glucose as glycogen. Now this process is helped by the insulin hormone. And when blood glucose levels are decreased, then glycogen is again converted back to glucose and it comes to the bloodstream. This process is facilitated by the hormone glucagon. Now, in cases when the individual is fasting or starving for a long time, then the reserved glycogen stores are also depleted and there is a shortfall of glucose. Therefore, what happens is that non-carbohydrate sources like proteins, fats, etc., they start generating glucose by a process known as gluconeogenesis. Now, this helps in maintaining the minimum or the basal level of glucose in the blood and therefore the proper functioning of the brain. Okay, so we will now see the starch or the glycogen upon the action of salivary amylase, it breaks down into disaccharides like maltose, sucrose, lactose, and further these disaccharides are acted upon by different amylolytic enzymes like maltase, sucrase, lactase, and these enzymes then convert the disaccharides into the monosaccharides from maltose to 2 glucose units, from sucrose to 1 glucose and 1 fructose, from lactose to 1 glucose and 1 galactose. Now, this is an enterocyte or an intestinal cell. Now, in this cell, we can see that the intestinal, this is the brush border which I had already described in the previous video. This is the brush border and here we will find several transporters, like this is the sodium glucose co-transporter, and this co-transporter proteins help in the absorption of glucose and galactose from the lumen, intestinal lumen, into the cell, and from there it ultimately goes to the portal vein and gets mixed in the bloodstream. Okay, so this sodium helps in the entry of glucose into the cell, like 1 glucose molecule from the intestinal lumen along with 2 sodium ions. And this SGLT does not transport fructose, it only transports sodium and glucose. Now this sodium is then excreted, like expelled out from the cell with the help of a sodium potassium ion exchanger, which means it expels 3 sodium ions and then it intakes 2 potassium ions. This also I have explained in the nerve and muscle unit. So with the help of an ATP hydrolysis, the sodium ion goes out and the potassium comes in. This sodium further helps in the co-transport of the glucose into the intestinal cells. And also there is a facilitative transporter called GLUT5 here. This GLUT5 can transport fructose but not glucose or galactose. So basically glucose, fructose and galactose are the simplest units of carbohydrates and they can be transported into the intestinal cells with the help of these hexose transporters. And GLUT1 and GLUT2, they GLUT2 can transport all the 3 like glucose, galactose and fructose. But GLUT1 can only transport glucose and galactose but not fructose. Similarly, GLUT3 can also transport glucose and galactose but not fructose. So these are present in other organs but mainly in the intestine. This GLUT2 is present and GLUT5 is present here. So with the help of these transporters, the glucose, galactose and fructose gets inside the bloodstream from the intestinal lumen and therefore they can be utilized by the body for various other activities. Next is the absorption of proteins. So, protein digestion leads to the following sequence. From protein, it is broken down to proteoses, then peptones, then polypeptides, then amino acids. Now, in the stomach, the very important enzyme is there, pepsin, which is secreted as pepsinogen, which is the inactive zymogen of the enzyme. And this is secreted by the chief cells of the stomach that we have already seen. And it is converted to pepsin in the acidic environment of the stomach, that is in presence of HCl. Now, milk present in the diet is coagulated by this enzyme called renin, which is present in high quantities in the infants, and this coagulated casein, like from caseinogen it converts to casein. Protein of milk is then hydrolyzed by pepsin. Now, this casein is capable of being hydrolyzed by the pepsin enzyme. And after passing through the stomach, obviously it reaches the small intestine, where there are two more important proteolytic enzymes. Those are trypsin and chymotrypsin. That is secreted by the pancreas, and it enters the duodenum via the pancreatic, the common duct. Now, the trypsin is secreted as trypsinogen. Similarly, a zymogen that needs to be activated by the action of another enzyme called enterokinase. Now, enterokinase activates this trypsinogen and converts it into active trypsin. So, trypsin recognizes and cleaves the peptide bonds adjacent to the C-terminal of arginine and lysine. So, it has a specific location where it goes and cleaves those bonds. Now, chymotrypsin is also secreted as chymotrypsinogen. So, you see that most of the enzymes are secreted as their inactive form, the zymogen form, and then they are further activated so that they can function properly. So, this in the presence of trypsin, the chymotrypsinogen is activated into chymotrypsin. Okay. Now, carboxypeptidase is another class of enzyme. It releases free amino acids from proteins from the C-terminal end. And a protein molecule, therefore a protein molecule is first cleaved by endopeptidase into smaller fragments, from the ends of which amino acids are hydrolyzed by exopeptidases. So, endopeptidase means the peptidase or the proteolytic enzymes that can cleave the protein molecule internally as well. And exo is that can cleave from the end of the protein molecule. So, the free amino acids are generated now by the help of all these different enzymes, and they are absorbed both by diffusion or by active transport in the intestine. So, there are 4 different transport systems. So, these are meant for neutral amino acids like alanine, glycine, dibasic amino acids like glycine, arginine, aspartic acid, glutamic acid, and also specific for glycine, proline, hydroxyproline. So, by the help of these 4 different transport systems, the absorptive cells of the intestine, they absorb the amino acids and they enter the blood, and from there it enters to the liver. Some of the amino acids are also moved along by coupling to sodium ion symport, similar to what we have seen in case of carbohydrate absorption. That also happens here, and sodium ion is import into the mucosal cells against their concentration gradient as well. So, besides amino acids, oligopeptides can also be transported along the intestinal epithelial cells by sodium symport. And large polypeptides which remain unhydrolyzed in the intestine are excreted from the body. So, as it reaches the large intestine, there is no other way to digest it, and therefore it has to be excreted out from the body. So, this sufficient amount of protein is also maintained in the bloodstream because the cell or the body needs to carry out different metabolic pathways and cellular processes. And amino acids that are not utilized therefore are marked for degradation, and they form the nitrogenous waste of the body. Okay. So, absorption of proteins, here you will see that there are polypeptides in the stomach, and by exocrine pancreas secretions, there is luminal digestion in the pepsin, trypsin, chymotrypsin. These all are helping here, and then free amino acids are generated 40% and oligopeptides are generated 60%. Again, the brush border enzymes are then coming into action, like endopeptidase and aminopeptidase. They are working on them, and therefore oligopeptides are also converted to the free free amino acids. Some oligopeptides are however converted into dipeptides and tripeptides, which by sodium symport enter the cells, and then dipeptidase or tripeptidase enzyme work on it and they convert them into free amino acids. And here, when the free amino acids are entering through the sodium symporter, they are directly getting inside the portal venous system. Okay, and this sodium is obviously expelled out from the cell with the help of a sodium potassium ion exchanger, and therefore the sodium can be further utilized for again symport of the amino acids. So this is very important that three kind of transporters can be there. One is uniporter, that only the one molecule will be passing through it in one direction only. And in case of symporter, like we have seen in sodium glucose, sodium amino acid, these are passing through of two molecules simultaneously at the same time in the same direction. Or there can be antiporters, which means one molecule is passing from one side to another side, in the opposite direction another molecule is passing from that side to this side. So these kinds of transporters are there, and there are several amino acid transporters as well. These are small nucleic acid transporter, then small neutral amino acid transporter, as well as large amino acid transporter. All these transporters are there. And in case of these oligopeptides, where the oligopeptides and hydrogen ions are co-transporting together inside the cell, then a peptidase for a dipeptidase works and then converts them into free amino acids. And then these free amino acids are again moving into the blood. Okay, so from the lumen, oligopeptides can be converted to amino acids, or they can be converted to dipeptides or tripeptides, and they can enter the intestinal cell, and from there also they can be converted to free amino acids for transport within the blood. The final part of absorption is the absorption of lipids. So in case of lipid absorption, first and foremost, the lipid molecules are broken down into fatty acids, glycerol, glyceride, in the presence of different lipases. There is gastric lipase, pancreatic lipase, intestinal lipase, all these are there. Now, only a small portion of fat is digested in the stomach with the help of gastric lipase. Majority of fat digestion is taking place in the intestine. That we already know. Now, when fat enters the small intestine, it stimulates the release of CCK hormone, the cholecystokinin. And this cholecystokinin helps in the stimulation of pancreas and contraction of the gallbladder to secrete bile into the small intestine. So when the bile comes into the duodenum, we know that bile contains a number of bile salts. Now these bile salts emulsify the fat into small globules called micelles. Now this micelle formation increases the surface area of the lipid molecule, and therefore it gives more area so that the pancreatic lipase can work on it. Now, some of the fatty acids can be directly absorbed by the intestine. The rest of the fatty acids, they can form micelles, and then they are absorbed through the crypts between the microvilli of the intestinal cells. The crypts I have already mentioned what are those, and from there the crypts, the cell can easily take up the micelles by the process of pinocytosis. Pinocytosis, we already know, it's a type of endocytosis which involves fluid molecules and very small fluid molecules ranging from 0.5 to 1 micron in diameter. And inside the cells, the long chain fatty acids recombine with triglycerides to form triglycerides, which are in the form of small globules called chylomicrons. Okay. Now this, if you can see that the bile salts are giving a layer of like a hydrophilic layer to the the bile salts are giving a layer so that the surface area is more, and therefore the hydrophilic head and hydrophobic tail part is not creating a problem. In case when the lipase is entering into the stomach or the intestine, it can easily work on this micelles. Now, chylomicrons are finally then transferred to the lymphatic vessels known as lacteals, and they absorb the proteins on their surface and they become chyle. So, chyle is a very slimy, milky fluid containing the chylomicrons along with the absorbed proteins on their surface, and therefore it helps in preventing the adhesion to each other. So, this is what I was explaining. So dietary fat, when it enters the stomach and intestine, the bile salts combined with it. So these are the smaller lipid droplets here. And therefore in the small intestine, we will see that there are lipid droplets as well as there are bile salts, and this micelles are being formed. And this lipid in the ingested food, this is insoluble in water. That we already know, the lipids are insoluble in water, and therefore they aggregate into large lipid droplets in the stomach. And the rate of digestion of these large lipid droplets is increased when they are broken down into very smaller droplets, number of smaller droplets. Therefore, what happens is that the surface area of the lipid droplets increase. When you convert a large lipid droplet to number of a few small lipid droplets, then the surface area is increased. Therefore, the lipase enzyme will have more surface to work on it. Now, this water insoluble fatty acids and monoglycerides are solubilized within the micelles formed by the bile emulsification. So, the bile salts help in this emulsification. The emulsification means the accessibility, the breaking down of the larger lipid droplets into smaller ones and giving access to the lipase to have more and more surface area to work upon. So, in this condition, the micelles are there, and therefore they are passing through the small intestine now. Yeah. And upon absorbing that, so water insoluble fatty acids and monoglycerides are solubilized within the micelles. Because the bile salts are giving them a layer of protection, they are solubilized within it, and therefore the emulsion is formed and emulsion is present. Now, they also contain lipid soluble vitamins, and then since the concentration of these constituents is larger in micelles than in aqueous solution, because in micelle it is very concentrated in a very small portion, small area. Therefore, the rate of delivery of fatty acids to the intestinal absorptive surface is much higher. So, with the help of micelles, more and more fatty acids and glycerols are being transported inside the intestine. Because in case of aqueous solution, it is very in a dilute condition, and in micelle it is concentrated in the center of the micelle. So therefore, it is getting absorbed very fast, at a faster rate. And once these monoglycerides and fatty acids leave the micelles, like here, they now diffuse through the membrane of the epithelial cells along their concentration gradient. So we know that in the inside of the intestinal cells, the concentration will be obviously lower, and within the lumen the concentration is higher. So it will obviously go towards this concentration gradient from the lumen to the inside of the intestinal cells, and therefore from there it will ultimately be carried on to a lacteal or to the lymph vessel or something like that, and therefore it will be ultimately carried into the blood vessel. So this is the intestinal epithelial cell. As you can see that the triglycerol and the fatty acids are going inside, and the chylomicrons are being formed. Chylomicrons are nothing but when they are entering inside the cell, then they recombine with glycerides and they again form triglycerides. And in the form of small globules, as you can see that the triglycerides are found in the form of small globules. This is known as chylomicrons. And when these chylomicrons are finally transferred to the lymphatic vessels or the lacteals, then they absorb water and other proteins and form a milky white fluid containing chylomicrons, proteins and etc. Then this is known as chyle. Okay. So this is basically the absorption of lipids ends here, and it is a very much hydrolytic process with the help of bile salts and different lipases. This digestion and absorption is possible, and this ends the unit 2 portion, that is of digestion, ends here. So please go through these videos, and if there is any confusion, then you can talk to me and ask me questions if you feel like it. Okay, thank you.